buckle up. We're about to shrink down and go full fantastic voyage into the microscopic world of the cell. From power-generating mitochondria to the selective fortress of the plasma membrane. Every structure has a story. And spoiler alert, it all matters for your AP Bio score. So whether you're the nucleus of your study group or just floating around like cytoplasm, this Unit 2 recap has you covered. Hey, I'm Melanie Kingett from The Absolute Recap, and I make AP Bio easier with podcasts, study guides, videos, the ultimate review packet, and the ultimate exam slayer. This updated Unit 2 review video, all about cell structure and function, follows the AP Biology course and exam description released by the College Board for fall 2025. But it's more than just prep for your next quiz. Think of it as your go-to tool for your midterm, your final, and of course, the AP Bio exam in May. Before we get started, don't forget to download your free copy of the AP Bio Ultimate Review Packet. The link's in the description. It's packed with a video-aligned study guide with an answer key, multiple choice practice, skill-building worksheets with video walkthroughs, and links to even more videos and podcasts. Not currently in AP Bio? No problem. These resources are great for any high school or college level biology course too. And if you're just here for a quick review, then check out the Ultimate Exam Slayer, focused 100% on exam prep. You'll get access to unit tests, full-length practice tests, one-pagers for every unit, math and graphing review sheets, and exclusive unit and exam tip videos. Okay, let's zoom in. 2.1 Cell Structure and Function. First up, ribosomes. These small but mighty structures are made of two key ingredients: ribosomal RNA, rRNA, and protein. Ribosomes are not membrane bound, which is an important distinction. You'll find them floating freely in the cytoplasm or attached to the rough endoplasmic reticulum. More on that in a bit. Why do they matter? Because ribosomes synthesize proteins based on instructions carried by messenger RNA. This process is called translation, and it's a central dogma of molecular biology. The fact that ribosomes are found in all forms of life, from bacteria to humans, supports the theory of common ancestry. Every living thing needs to make proteins, and ribosomes make that possible. Next, let's look at the endomembrane system, a group of membrane-bound organelles that work together like a cellular assembly line to modify, package, and transport within the cell and beyond. Here's the breakdown. The endoplasmic reticulum, ER, comes in two varieties. Rough ER has ribosomes attached. It helps compartmentalize the cell and plays a major role in protein synthesis and processing. Smooth ER, on the other hand, has no ribosomes. It handles lipid synthesis and helps detoxify substances in the cell. Think of it as the cellular version of a detox spa. The Golgi complex consists of flattened sacs that fold, chemically modify, and package proteins for their final destination. This could be within the cell or out to the extracellular environment. The system also includes the nuclear envelope, plasma membrane, lysosomes, transport vesicles, and vacuoles, all working together to keep the traffic of materials flowing smoothly. Let's talk about waste and storage, because even cells need a cleanup crew. Lysosomes are membrane-bound sacs filled with hydrolytic enzymes that break down damaged cell parts, waste, and even pathogens. They also play a key role in apoptosis, or programmed cell death, which is essential for development and disease prevention. Vacuoles are membrane-enclosed storage units with different functions depending on the type of cell. In plant cells, the central vacuole is huge and crucial. It stores water and nutrients and maintains turgor pressure, helping the plant stay upright. In animal cells, vacuoles are smaller and more numerous, storing ions, nutrients, and waste products. Now for some serious energy talk. The mitochondria. These double-membraned organelles are the site of aerobic cellular respiration. The outer membrane is smooth, but the inner membrane is highly folded into structures called cristae. This folding increases surface area, allowing for more ATP production, the energy currency of the cell. The compartments created by these membranes allow different metabolic reactions to occur in an organized, efficient way. So more reactions equals more energy and more function. Last but not least, chloroplasts. These are found only in plants and photosynthetic algae and are the site of photosynthesis. Like mitochondria, chloroplasts have a double membrane, and inside they contain stacks of membranes called thylakoids, where light energy gets converted into chemical energy, glucose. Just like mitochondria, chloroplasts create compartments to increase efficiency. Got all that? I know, there are a lot of organelles to understand. I highly recommend trying the Unit 2 practice sheet and video inside the Ultimate Review Packet to sharpen your organelle identification skills by structure and function. 2.2 Cell size. What does surface area and volume actually mean for cells? Surface area refers to the total area of the cell's outer membrane, the interface between the inside of the cell and the environment. Volume is the amount of space inside the cell, basically the cell's stuff, or cytoplasm and organelles. For cells to survive, the surface area has to be big enough to support the needs of volume inside, like supplying nutrients and removing waste. Here's the catch. As cells get bigger, its volume grows much faster than its surface area. This means the surface area to volume ratio actually decreases, and that's a problem. A smaller ratio means less membrane surface to exchange materials per unit of volume inside the cell. So the cell can't get what it needs or get rid of waste efficiently. That's why cells are small, easier to keep a high surface area to volume ratio and stay efficient. If cells get too big, they'd starve internally or get poisoned by waste buildup because their membranes just can't keep up. Some cells and organelles solve this by having lots of folds or extensions in their membranes, increasing surface area without making the structure huge. But the surface area to volume ratio doesn't just affect single cells, it affects whole organisms too. As animals get bigger, their surface area to volume ratio decreases, which changes how quickly they gain or lose heat. Smaller animals lose heat quickly and have higher metabolic rates to stay warm, while bigger animals hold heat longer and tend to have slower metabolisms. This explains why a tiny shrew's heart beats faster and it eats constantly, while an elephant's metabolism is much slower. Surface area and volume calculations help biologists predict how cell size affects function and efficiency. All of the equations you need are on the formula sheet, and if you need help understanding the math, check out the math review sheet in the Ultimate Exam Slayer. 2.3 Plasma membrane. At the heart of the cell membrane is the phospholipid bilayer, a double layer of molecules that have split personalities. Each phospholipid has a hydrophilic head, meaning it loves water, and hydrophobic tails that avoid water at all costs. These molecules arrange themselves with their heads facing the watery environments inside and outside the cell, while the fatty acid tails huddle together in the middle, away from water. This setup forms a flexible but protective barrier that's selective about what crosses through. But the membrane isn't just made of lipids. Scattered throughout are proteins. Some float, some anchor, and each one plays a unique role. These proteins can be hydrophilic, hydrophobic, or a combination of both, depending on the side chains of their amino acids. These proteins are involved in transport, signaling, and maintaining structure. So why is it called a fluid mosaic model? Fluid refers to the fact that the membrane components aren't locked in place. They move and shift within the layer. Mosaic refers to the variety of components embedded in the membrane: phospholipids, proteins, cholesterol, glycoproteins, and glycolipids, forming a complex, colorful patchwork. 2.4 Membrane permeability. The plasma membrane's job is to separate the internal environment of the cell from the outside world, and it does so with selective permeability. So what actually makes it pass? Small, nonpolar molecules like nitrogen gas, oxygen, and carbon dioxide slip right through, no problem. But larger polar molecules or ions, they need assistance, and that's where embedded channels and transport proteins come in. Now, the plasma membrane isn't working alone. Some cells have an extra layer of security, the cell wall. Found in bacteria, archaea, fungi, some protists, and plants, the cell wall provides a sturdy structural boundary. Importantly, it protects against osmotic lysis, that dangerous situation when too much water rushes in and the cell could literally burst. 2.5 Membrane transport. Selective permeability is essential because it allows cells to create concentration gradients, differences in the amount of a substance inside versus outside the cell. Without those gradients, cells couldn't power many of the processes that keep life going. Now let's talk about how molecules actually move across the selective barrier. The first method is passive transport. Think of it like rolling a ball downhill. Molecules naturally move from areas of high concentration to areas of low concentration, and the cell doesn't have to spend any energy. Diffusion, facilitated diffusion, and osmosis all fall into this category. But sometimes cells need to move molecules against the flow, like pushing a ball uphill. That's where active transport comes in. Active transport requires energy, usually in the form of ATP, because it's moving substances from low concentration to high concentration, which doesn't happen naturally. So what about molecules that are simply too big to squeeze through the membrane? No matter what. That's when cells rely on bulk transport, endocytosis and exocytosis. In endocytosis, the cell wraps its plasma membrane around the material from the outside, creating little vesicles to pull it in. In exocytosis, it's the opposite. Vesicles inside the cell fuse with the plasma membrane and release their contents out into the environment. 2.6 Facilitated diffusion. Facilitated diffusion is like a special entrance that helps certain molecules passively get across the membrane. Think of it like a toll-free bridge. You're still going downhill, but you need a structure to cross safely. For charged ions, the membrane is a total barrier without channel proteins. These ions rely on proteins that act like tunnels carved straight through the lipid bilayer. As they move, they can change the electrical balance across the membrane. We call that polarization. If you've ever rubbed a balloon on your hair and stuck it to the wall, you've seen static electricity in action. Cells use a similar trick with ions to create charges across their membranes. Now, what about water? It may seem small, but moving lots of water at once isn't so simple. That's why cells use aquaporins, protein channels dedicated to transporting huge amounts of water quickly and efficiently. 2.7 Tonicity and osmoregulation. When it comes to water movement in and out of cells, the external environment makes all the difference. A solution can be hypotonic with less solute concentration, hypertonic with greater solute concentration, or isotonic compared to the inside of the cell. The rule of thumb here is simple. Water always moves from hypotonic to hypertonic regions. Or, if you like to think of it in terms of potential, water flows downhill from areas of high water potential to areas of low water potential. This process, osmosis, is passive and doesn't require energy. But living organisms aren't just passive bystanders. They've got to regulate water and solute balance to survive. That's where osmoregulation comes in, the body's way of making sure cells don't shrivel up like raisins or burst like overfilled balloons. Osmoregulation helps maintain growth and homeostasis by carefully controlling solute composition in water potential inside cells. The equations for water potential and solute potential are on the equations and formula sheet provided on the exam. A higher solute concentration means lower solute potential. Basically, the saltier it gets, the less water potential there is. And calculated negative values for solute potential totally makes sense, because adding solutes makes water potential drop. If you need more help with these calculations, you'll find it in the Unit 2 practice sheet in the URP and math practice sheet in the UES. 2.8 Mechanisms of transport. When it comes to moving molecules and ions across the cell membrane, sometimes simple diffusion isn't enough, and that's where active transport comes in. Without it, larger charged molecules couldn't get where they need to go, and the whole system would stall. Membrane proteins decide what gets transported, and they use energy from ATP to push molecules against their concentration gradient. One of the most famous of these transport proteins is the sodium-potassium pump. For every cycle, 3 sodium ions are pushed out of the cell and 2 potassium ions are brought in. This uneven exchange creates and maintains what we call a membrane potential, an electrical difference across the membrane. You can picture this like charging a battery. The pump sets up an electrochemical gradient, storing potential energy that the cell can use later, just like your phone stores charge until you need it. And ATP, that's the currency funding the whole operation. 2.9 Cell compartmentalization. If you tried to chop vegetables, boil pasta, and bake cookies all on the same counter at the same time, things would get messy fast. But when you use separate cutting boards, pots, and ovens, each task happens in its own space without interfering with the others. And that's exactly what compartmentalization does in cells. It keeps reactions organized and efficient. Internal membranes also solve another big problem, space. By folding and stretching out into complex shapes, they massively increase surface area. More surface area means more places for enzymes to work and reactions to happen. Mitochondria, for example, pack their inner membranes with folds of cristae, giving them the surface area needed to churn out lots of ATP. 2.10 Origins of cell compartmentalization. When we look at the big differences between prokaryotic and eukaryotic cells, one of the biggest game changers is the presence of membrane-bound organelles. Eukaryotic cells have these separate compartments, while prokaryotes like bacteria generally do not. But here's the twist. Some of those eukaryotic organelles actually started out as their own free-living prokaryotic cells, based on their own DNA, ribosomes, and double membranes. The leading explanation formation is called the endosymbiotic theory. Imagine one cell swallowing another, not to digest it, but to form a partnership. Over time, the engulfed prokaryote became a permanent resident, evolving into organelles like mitochondria and chloroplasts. Meanwhile, prokaryotic cells might not have these separate membrane rooms, but they're not just a shapeless blob. They still have organized internal regions that carry out specialized functions, like the nucleoid, where DNA hangs out, or thylakoid membranes in cyanobacteria, which help capture light energy. To recap, all living things are made of cells, prokaryotic or eukaryotic. Organelles, like the nucleus, mitochondria, and chloroplasts, carry out essential cell functions. The endomembrane system coordinates protein production and transport. The plasma membrane is selectively permeable thanks to phospholipids and proteins. Diffusion, osmosis, and active transport move materials across membranes. Surface area to volume ratio affects how efficient a cell can be. Compartmentalization lets eukaryotic cells multitask efficiently. Mitochondria and chloroplasts have their own DNA, supporting the endosymbiotic theory. Overall, understanding cell structure helps explain how life functions on every level. Need more support? Sign up for the Ultimate Review Packet. The Unit 2 free preview includes the study guide and practice multiple choice questions. And if you're ready to shift into full exam mode, the Ultimate Exam Slayer has practice tests, one-pagers, and tips to help you dominate in May. All links are in the description. Thanks for watching, and I'll see you next recap for Unit 3, Cellular Energetics.